Now it's important to note that this is step 1, it is irreversible. This is step 3, it is irreversible. This is reversible, this is again reversible, and this is reversible. Dihydroxyacetone phosphate, if it has to enter the second phase of glycolysis, then it has to be converted to glyceraldehyde 3-phosphate. So it is clear that these two compounds are formed, but dihydroxyacetone phosphate is finally converted to glyceraldehyde 3-phosphate. Now we'll take the first step, glucose converted to glucose 6-phosphate. This is irreversible. This is rate limiting step. That is, it is one of the slowest step. And this is irreversible step. This is rate limiting step. That is the slowest step. Now we'll discuss the hexokinase enzyme. It's very important to know that hexokinase exists in 4 forms. Hexokinase 1, 2, 3 and 4. These are also known as A, B, C and D. Hexokinase 1 is found in brain, kidney and adipose tissue. Hexokinase 2 is found in skeletal muscles. Hexokinase 3 is found in adipose tissue. And hexokinase 4 is found in liver. It's important to note that first is also found in liver, second is also found in liver, third one is also found in liver, but this is found only in liver. This is termed as glucokinase. So glucokinase is basically the hexokinase 4. Now we'll compare the hexokinase with glucokinase. That is, it is 1, 2, 3 and that one is 4. It is found in all tissues, in liver only. Phosphorylation of glucose, fructose, mannose. This is involved in phosphorylation of glucose only. Km value, it is low, it is high. That is 0.1 millimole and it is 10 millimole. Low Km value indicates high affinity, while high Km value indicates low affinity. Inhibition by glucose 6-phosphate. Yes. No. This type of inhibition is allosteric inhibition. Then is stimulated by insulin. No. Yes. So these are the various differences. So we can say that hexokinase basically ensures the supply of glucose to the cells when it requires, while the glucokinase drives away the extra glucose from the blood whenever it is in excess, irrespective of that whether the cell requires it or not. Now the next is phosphohexoisomerase. But before that, glucose 6-phosphate is formed. We have to discuss some points about that. It is impermeable to cell membrane. Phosphorylation promotes the product or intermediate to enter in other processes or metabolisms. Glucose, when phosphorylated, it acquires negative charge and it becomes impermeable to the cell membrane. Glucose is also substrate for other metabolic pathways like glycolysis, gluconeogenesis, glycogen synthesis and HMP shunt. Then phosphoisomerase, fructose 6-phosphate is formed. This is isomerization. Then next step, fructose 6-phosphate to fructose 1,6-bisphosphate by phosphofructokinase. It is basically PFK1. This step is also irreversible. This step is also a rate limiting step. And most important is that it is the first committed step of glycolysis, after completion of which the intermediate will complete the glycolysis. It will not go in any other reaction. Now next step is the breakdown of fructose 1,6-bisphosphate by the aldolase enzyme. Aldolase enzymes are also having three types, A, B and C. Three forms are there. A is mostly, it is found in most of tissues. It is most common. B is found in silk. What is that? That is stomach, intestine, liver and kidney. While the aldolase C is found in heart, ovary and brain. So finally, the compounds glycerylate 3-phosphate and dihydroxyacetone phosphate are formed. So we can conclude that after first phase of glycolysis, one molecule of glucose is converted to 2 molecules of glycerylate 3-phosphate with use of 2 ATPs. In next lecture, we'll discuss the second phase of glycolysis. Hit the like button, share and subscribe our channel to get the latest updates and notifications.